Hello everyone, I am Subalakshmi. Welcome all of you to this channel. In this video, I am going to discuss about 20 multiple choice question on the topic of classification and cell structure of fungi with their proper explanation. So before we get into the discussion, let's start from a beautiful message. Be a believer and you will be an achiever. So believe in yourself and the universe will open doors where you once saw the walls. Faith fuels action and the actions leads to achievement. When you trust your inner strength, challenges become stepping stones. Let your belief be unshakable and success will follow effortlessly. Okay, see the first question. In which group of fungi the mycelium in the vegetative phase usually lacks any kind of internal partitions? Option A Deuteromycetes, option B Basidiomycetes, option C Phycomycetes, option D Ascomycetes. Here the correct answer is option C, that is Phycomycetes. We know Phycomycetes, also called algal fungi, and the members are considered as lower fungi, okay, found in aquatic and on decaying wood in moist and damp places, or as obligate parasites on plant. Somatic or vegetative phase consists of either unicellular thallus or a non-septate sinusoidal mycelium. So, this group of fungi usually lacks any kind of internal partition. Okay, so here the correct answer is option C. And here in other group of fungi, that is in Deuteromycetes, Basidiomycetes and Ascomycetes, internal partitions are present, means their mycelia are septate. Okay. Then next question, the suggested formula for fungal chitin. Here the correct answer is option D. We know the composition of cell wall is variable among the different groups of fungi or between the different species of the same group. In the majority of fungi, the wall lacks cellulose but contains a form of chitin known as the fungus cellulose, and the suggested formula for fungus chitin is C22, H54, N4, O21. Okay, so here the correct answer is option D. Then next question, the first detailed classification of fungi was proposed by which mycologist in his 3-volume book Systema Mycologicum? Option A E. M. Fries, option B K. C. Mehta, option C E. J. Butler, option D Ainsworth. Here the correct answer is option A, that is E. M. Fries. E. M. Fries refers to Elias Magnus Fries in his 3-volume book, that is Systema Mycologicum, published during 1821 to 1832. He divided fungi into 4 classes, that is Coniomycetes, Hyphomycetes, Gastromycetes and Hymenomycetes. And remember, he was a mycologist and botanist. For his contribution, he is sometimes called the Linnaeus of mycology. Okay. Then see the other option, that is K. C. Mehta, refers to Karamchand Mehta, known for his contribution to plant pathology, particularly his research on rust disease in India. Then in the option C, that is E. J. Butler, refers to Sir Edwin Johann Butler, was an Irish mycologist and plant pathologist. And remember, E. J. Butler is known as father of Indian mycology and plant pathology. Then in the option D, that is Ainsworth, he was a British mycologist and scientific historian. He proposed a more natural system of classification of fungi. Okay. Then next question, the invaginations of cell membrane in fungi is either in the form of an infolded convoluted pocket or a pouch enclosing granular or vesicular material called option A lamellosome, option B mesosome, option C lomosome, option D microsome. Here the correct answer is option C, that is lomosome. So when you see the structure of cell in fungi, the cell membrane is a delicate, extremely thin living membrane which closely invests the protoplast. The cell membrane or plasma membrane is pressed against the cell or hyphal wall except for occasional invaginations in some regions. The invagination is either in the form of an infolded convoluted pocket or a pouch enclosing granular or vesicular material. Two mycologists, Murray and MC Lear, named it lomazome. Okay, so here the correct answer is option C. Then see the other option, that is lamellazome. Lamellazome is a membranous, coiled, semicircular structure attached to the plasma membrane found in blue-green algae and is involved in photosynthesis. Then in the option B, that is mesosome. Mesosome is an infolding of the plasma membrane in prokaryotic cells appearing as vesicles or lamella and is involved in process like DNA replication, cell division and respiration also. Then in the option D, that is microsomes. Microsomes are vesicle-like structure formed from fragments of the endoplasmic reticulum during tissue homogenization used to study functions of the endoplasmic reticulum and drug metabolism. Okay. Then question number 5, Gaine, Bohugan and Barnage recognized only 4 classes, that is Phycomycetes, Ascomycetes, Basidiomycetes and Deuteromycetes on the basis of option A, reproductive characters and thallus structure, option B, structure of mycelium and characteristic of spores, option C, life cycle and development of fructification, option D, thallus structure and life cycle. Here the correct answer is option B, that is structure of mycelium and characteristic of spores. So you can see here that is Gaine, Bohugan and Barnage recognized only 4 classes of fungi, that is Phycomycetes, Ascomycetes, Basidiomycetes and Deuteromycetes. So they mostly focused on the structure of mycelium and characteristic of spores for the classification of fungi. For example, in Phycomycetes, mycelium aseptate and multinucleate. Sexual spores are oospores and zygospores. Then in Ascomycetes, mycelium fairly branched and septate. Septa are simple. Characteristic spores are endogenously produced ascospores. Then in the Basidiomycetes, mycelium branched and septate, septa with dollipores. Characteristic spores are exogenously produced basidiospores. Then in the Deuteromycetes, mycelium branched and septate, sexual spores are absent. Okay. So here the correct answer is option B. Then next question, the fungi with mycelium branched and septate and the characteristic spores are exogenously produced belongs to which class? Option A, Ascomycetes, option B, Phycomycetes, option C, Deuteromycetes, option D, Basidiomycetes. Here the correct answer is option D, that is Basidiomycetes. We just discussed about that is in Basidiomycetes, mycelium branched and septate, septa with dollipores. Characteristic spores are exogenously produced basidiospores. Okay, so here the correct answer is option D. Then in the other option, that is in Ascomycetes, mycelium fairly branched and septate, but the characteristic spores are endogenously produced, not exogenously. Okay. Then in Phycomycetes, mycelium aseptate and multinucleate. Then in Deuteromycetes, mycelium branched and septate, but here the sexual spores are absent. Okay. Then next question, according to the International Rule of Botanical Nomenclature, what is the suffix of the names of fungal subclasses? Option A, Mycetidae, option B, Mycota, option C, Mycotina, option D, Mycetes. Here the correct answer is option A, that is Mycetidae. We know the Committee on the International Rule of Botanical Nomenclature has made the following recommendation, that is the names of division of fungi should end in Mycota. The subdivision should end in mycotina. The names of classes should have a suffix, that is mycetes. The subclasses should end with a suffix, that is mycetidae, and the names of orders should end in else. Then the families should end with a suffix, that is sc. Okay. So here the correct answer is option A, that is mycetidae is the suffix of the names of the fungal subclasses. Then mycota is the suffix of the names division of the fungi, and mycotina is the suffix of subdivision of the fungi, and last one, that is mycetes. Mycetes is the suffix of the classes. Okay. Then next question, a more natural system of classification of fungi was proposed by option A Alexopoulos, option B Anton de Berry, option C Ainsworth, option D Antonio Micheli. Here the correct answer is option C, that is Ainsworth. So here a more natural system of classification of fungi was proposed by Ainsworth. Ainsworth placed all the fungi in the kingdom Mycota. Again, he divided the kingdom Mycota into two divisions, that is Myxomycota and Eumycota. Okay. Again, he divided the division Myxomycota directly into four classes. These are Acrasiomycetes, Hydroxymyxomycetes, then Myxomycetes, and last one is the Plasmodiophoromycetes. But the second division, that is Eumycota, divided into 5 subdivisions, that is Mastigomycotina, Ascomycotina, Deuteromycotina, Zygomycotina and Basidiomycotina. Again, the subdivision Mastigomycotina divided into 3 classes, that is Chytridiomycetes, Hypochytridiomycetes and Oomycetes. Similarly, Ascomycotina divided into 6 classes, that is Hemiascomycetes, Loculoascomycetes, Plectomycetes, Labulobeniomycetes, Pyrenomycetes and Discomycetes. Again, the subdivision Deuteromycotina divided into 3 classes, that is Blastomycetes, Coelomycetes and Hyphomycetes. Then Zygomycotina divided into 2 classes, that is Zygomycetes and Trichomycetes. Similarly, the last one, that is Basidiomycotina, divided into 3 classes, that is Teleomycetes, Hymenomycetes and Gastromycetes. Okay. So this is the classification system proposed by Ainsworth, and remember, this classification is based on morphology, especially of reproductive structure of fungi, and he includes fungi along with slime mold under the kingdom Mycota, right? Then see the other option, that is Alexopoulos. Alexopoulos placed fungi in a separate kingdom, that is Myceti, and hence the study of fungi is known as mycetology. Then in the option B, that is Anton de Berry. He was a German botanist, microbiologist and mycologist also. He is considered a founding father of plant pathology as well as the founder of modern mycology. So he is known as the father of modern mycology and plant pathology. Okay. Then in the option D, that is Antonio Micheli. He was a noted Italian botanist and he was the first person to attempt the scientific study of fungi and gave accurate description of fungi in his book Nova Plantarum Genera, and he is known as the father of mycology. Okay. Then next question, the fungus associated with the discovery of gibberellin is option A Fusarium oxysporum, option B Fusarium moniliforme, option C Fusarium longipes, option D Fusarium solani. Here the correct answer is option B, that is Fusarium moniliforme. We know the plant hormone gibberellin was discovered in 1926 by Japanese botanist Ichi Kurosawa while investigating the foliage seedling or Bakena disease in rice caused by the fungus Gibberella fusarium, also known as the Fusarium moniliforme. So here the correct answer is option B. Then next question, which one of the following is called fungi imperfecti? Option A basidiomycetes, option B ascomycetes, option C oomycetes, option D deuteromycetes. Here the correct answer is option D, that is deuteromycetes, also called fungi imperfecti. Why? Because the defining characteristic of deuteromycetes is that their sexual phase or perfect stage is either absent or unknown. Okay. So the fungal class, that is deuteromycetes, also called fungi imperfecti. Okay. So here the correct answer is option D. And remember, basidiomycetes are also called clove fungi, ascomycetes are commonly called sac fungi, and phycomycetes are also called lower fungi or algal fungi. Okay. Then next question, ascocarp in discomycetes is option A perithecium, option B acervulus, option C apothecium, option D cleistothecium. Here the correct answer is option C, that is apothecium. So we know ascocarp is the fruiting body of an ascomycetes fungus, and we can see here that discomycetes is one of the class of the subdivision Ascomycotina, and remember, in the class discomycetes the ascocarp also known as apothecium. And what is apothecium? Apothecium is a cup-shaped fruiting body, and the asci are arranged in orderly layer on this exposed surface, allowing for easy spore dispersal. Okay, so here the correct answer is option C. Then see the other option, that is perithecium. Perithecium is a flag-shaped fruiting body in certain ascomycetes fungi containing asci and typically opening via a pore. Then in the option B, that is acervulus. Acervulus is a asexual fruiting body produced by certain fungi, particularly those that are parasitic on plants. Then in the option D, that is cleistothecium. Cleistothecium is a closed spherical fruiting body in some ascomycetes fungi from which spores are released only by decay or disintegration, right? Then next question, Dollipore septum is characteristic of members of option A Uridinales, option B Pagigales, option C Agaricales, option D Saprolegniales. Here the correct answer is option C, that is Agaricales. We know Dollipore septum is a characteristic feature of Basidiomycetes, right? But remember, they are not universally present in all members of this group. For example, Dollipore septum absent in rust and smut fungi, right? Rust fungi are classified within the order Uridinales, and smut fungi are classified within the order Ostilaginales. And remember, in the members of these two order, Dollipore septum generally absent, right? So here option A is the incorrect. Then in the option B, the order Pagigales. We know the order Pagigales present within the phylum Ascomycota, and we know Dollipore septum not seen in this group. Okay, so option B is also incorrect. Then in the option D, that is Saprolegniales. We know Saprolegniales present within the phylum Oomycota, and we know Dollipore septum is not the characteristic feature of Ascomycota and Oomycota. Then in the option C, that is Agaricales. So we know the order Agaricales present within the phylum Basidiomycota, in which Dollipore septum are generally present. Okay, so here the correct answer is option C. Then next question, in Dollipore septum of Basidiomycetes, on both the sides of the dollipore, a double membranous structure, septal pore cap is present called option A apressorium, option B parenthosome, option C lamellosome, option D mesosome. Here the correct answer is option B, that is parenthosome. When you see the detailed structure of the Dollipore septum, it is composed of a pore cap surrounding a septal swelling and a septal pore, right? This is septal swelling. A prominent barrel shaped swelling surrounds the central pore, right? Then the central pore allows for the passage of cytoplasm and small organelles between cells. Then the septal pore caps, also called parenthosome, which is a specialized membranous resembling parenthesis like a cap each side of the pore. Then the two main functions of polypore septum are intercellular communication and regulation of nuclear movement. Then next question, lysergic acid diethyl amide is obtained from option A Clavacia, option B Trichoderma, option C Ascobulus, option D Claviceps. Here the correct answer is option D, that is Claviceps. LSD or lysergic acid diethyl amide is synthesized from lysergic acid and the ergot fungus, specifically Claviceps purpura, contains lysergic acid which is a precursor of LSD. And remember, LSD is a derivative of lysergic acid and it's synthesized from lysergic acid through a chemical process, not directly extracted from the fungus. In 1938, Albert Hofmann synthesized LSD while researching the lysergic acid derivatives. And remember, Claviceps purpura is a parasitic fungus that grows on certain types of grasses, causes the ergot disease. Then see the other option, that is Clavacia. Clavacia is a genus of puffball mushrooms. It is generally found in temperate areas throughout the world. Then in the option B, that is Trichoderma. Trichoderma is a genus of fungi that is present in soils where they are the most prevalent culturable fungi. Then in the option C, that is Ascobulus. Ascobulus, it is a coprophilous fungus that is grows on animal dung. Okay. Then question number 15. In fungus, the pore is lined with a ring of electron dense material known as option A ornine body, option B somatic cyst, option C mucilaginous body, option D chitinous body. Option D chitinous body. Here the correct answer is option A, that is ornine body. Ornine bodies are specialized organelles, specifically a type of peroxisome that are found in filamentous fungi. They are named after the Russian mycologist Michael Stefanovich Ornin. So it is called ornine body. Okay. Then what are the function? Their primary function is to seal the septal pores, which are openings in the cell wall between fungal hyphae in response to injury or stress. Right? This prevents the loss of cytoplasm and allows the cell to repair the damaged plasma membrane. Then where these are located? Generally, these are located near the septa, means the walls that divide the hyphal compartment in filamentous fungi, and ornine bodies are composed of a dense proteinaceous core surrounded by a membrane. Okay, so in fungus the pore is lined with a ring of electron dense material known as ornine body. Then next question, who included bacteria and slime molds in fungi in his famous book Xylose Fungorum and divided fungi into 6 classes? Option A Martin, option B Gauman, option C Sacrodo, option D Besse. Here the correct answer is option C, that is Sacrodo. So the scientist Sacrodo included bacteria and slime molds in fungi in his famous book, that is Xylose Fungorum, and divided fungi into 6 classes. These are Schizomycetes, Myxomycetes, Phycomycetes, Ascomycetes, Basidiomycetes and Deuteromycetes. Okay. Then see the other option, that is Martin. Martin system of classification based on the septation of the mycelium and spore characters. He divide fungi into two groups, that is Myxomycetes and Eumycetes. In Myxomycetes, plant body in the form of amoeboid naked protoplast, and in Eumycetes, the plant body unicellular or multicellular filamentous. Again, Eumycetes divided into four classes, that is Phycomycetes, Ascomycetes, Deuteromycetes and Basidiomycetes. Then in the option B, that is Gowman. Two mycologists, Gowman and Dodge, proposed a system of classification based on the life cycle and development of fructification. According to them, fungi are divided into four classes, that is Archimycetes, Phycomycetes, Ascomycetes and Basidiomycetes. Then in the option D, that is Bese. Bese's system of classification was largely based on reproductive characters, and he divides fungi into two groups, that is lower fungi and higher fungi. In lower fungi, fruiting bodies are not formed, and he include one class, that is Phycomycetes, under the group lower fungi. And in higher fungi, fruiting bodies are formed, and he divide higher fungi into three classes, that is Ascomycetes, Basidiomycetes and Deuteromycetes. Okay. Then next question, Sabrod media for the growth of fungi is composed of option A glucose and ammonia, option B dextrose and peptone, option C sucrose and maltose, option D glucose and fructose. Here the correct answer is option B, that is dextrose and peptone. Remember, Sabrod media, also called SDA media, means Sabrod dextrose agar medium, right? It was developed by Raymond Sabrod. He used it for cultivating and isolating fungi, especially dermatophytes and yeast. Dermatophytes means those fungi that cause skin, hair and nail infection. So these are basic composition of Sabrod media, that is peptones. Peptones provide a source of nitrogen and amino acids for fungal growth. Then dextrose serves as a carbon and energy source. Then agar acts as a solidifying agent. Last one is the low pH. The low pH inhibit the growth of many bacteria, making it selective for fungi. Okay, so here the correct answer is option B, that is the Sabrod media for the growth of fungi is composed of dextrose and peptone. Okay. Then next question, incorrect statement is option A, chitin wall of most fungi is permeable both to water and substance in true solution. Yes, it is a correct statement. Then in the option B, in the majority of fungi, the cell wall lacks cellulose. It is also a correct statement. We know in the majority of fungi, the cell wall lacks cellulose, and the cell wall contains a form of chitin known as fungus cellulose. Okay. Then in the option D, the basic structural constituent of the cell wall in the Zygomycetes and higher fungi is chitin. Yes, it is also a correct statement. But in the option C, that is chitin is a polysaccharide based on the phosphorus containing sugar. No, it is a incorrect statement. Why? Because chitin is a polysaccharide based on the nitrogen containing sugar, not phosphorus containing sugar. Okay. And remember, the suggested formula for fungal chitin is C22H54 N4O21. Okay. So chitin is a linear polysaccharide, meaning it's a long chain of repeating sugar unit. Which sugar unit? Specifically N-acetyl glucosamine, also known as N-acetyl D-glucosamine. Okay. Then the linkage, the N-acetyl glucosamine monomers are linked together by beta-1,4 glycosidic bond. Okay. So here option C is the incorrect one, because chitin is a polysaccharide based on the nitrogen containing sugar, not phosphorus containing sugar. Okay. Then next question, the fungus which is commonly known as Drosophila of the plant kingdom, belongs to which class? Option A Ascomycetes, option B Basidiomycetes, option C Deuteromycetes, option D Phycomycetes. Here the correct answer is option A, that is Ascomycetes. And remember, the fungus Neurospora is known as the Drosophila of the plant kingdom. Why it is called Drosophila of the plant kingdom? Because like Drosophila in the animal kingdom, it is widely used as a model organism for genetics and biochemical studies. So Neurospora is a genus of Ascomycetes fungi. Commonly it is called red or pink mold. And remember another important point, that is one gene one enzyme hypothesis proposed by Biddle and Tatum when they work on the fungi Neurospora. Okay, so Neurospora belongs to Ascomycetes class of the fungi. Okay, then last question, correct statement is, in the class Chytridiomycetes of the lower fungi is furnished with two whiplash flagella. No, it is incorrect one. Remember, the motile cell in the class Chytridiomycetes of the lower fungi is furnished with single whiplash flagellum with a pointed tip, like in the figure A. Okay, so in the class Chytridiomycetes, a single whiplash flagellum is present, not two. Okay, then in the option B, the representatives of the class Hypochytridiomycetes possess a single flagellum of whiplash type. No, it is also an incorrect statement, because the representative of the class Hypochytridiomycetes possess a single flagellum, a tin cell type. Okay, then in the option C, the higher fungi and Zygomycetes have flagella. It is also an incorrect statement, because the higher fungi and Zygomycetes lack flagella. Then in the last one, that is biflagellate motile cells are characteristic of the class Oomycetes. It is a correct statement, because in the motile cells of the class Oomycetes with a pyriform shape, the two flagella are inserted at the anterior end. Okay, so option D here is a correct statement. So in this video I covered all the 20 multiple choice questions with their proper explanation. So thank you.